Thank you so very much for the invitation and the huge honour to be here. I've never quite had as many nerves and as much honour at the same time, but thank you. So I'm going to give you a bit of a flavour of the work that I and my team do on genomics. And I should admit, I'm not really a card-carrying geneticist. There's some scientists in the audience who I'm sure are far better geneticists than I am. But I'm really excited about the potential for genomics to inform how we improve care of people at risk of and living with diabetes in the future. And that's really what underpins the work that I do. So I'm going to give you a flavour of all of that this evening. Some conflicts of interest, which are mostly held by the Genes and Health study that I'm going to talk to you about, which receives funding from a number of sources, including some pharmaceutical companies, but no direct funding to myself. I would ask you please to not take photos because I'm going to be presenting quite a lot of unpublished data this evening. So I thought I'd start by giving you just a little flavour of where I'm based in East London. And I look eastwards a lot in my life, to East London, but also to South Asia. And I work in a very diverse population with some really huge and wonderful South Asian influences, including knowing exactly when the Indian mango season has started in my diabetes clinic, because there's a lot of mangoes imported to East London, thankfully. And there's also some great restaurants that my son and I enjoy very regularly. Dr.Anjana, you did invite lots of conversations about food yesterday, so I thought I would continue. So looking further east, beyond East London, is something I've done really since the start of my career as a diabetologist. And some pieces of work that really inspired me very early on were research and this famous image that I'm sure many of you know of two great stalwarts in the diabetes research field, one of whom, John Yudkin, was my very first and terrifying boss, really just demonstrating that two men, one of white European origin and one of South Asian origin with the same BMI, had vastly different body fat proportions and distribution on DEXA scan. And this is work, of course, that has been taken much further by my fantastic colleagues at MDRF. During my PhD, I was very lucky to spend some time in Bangladesh and work with colleagues there who were following on from the Bangladeshi famine in the 1970s and studying the long-term impact and increased risk of type 2 diabetes in individuals exposed to famine. And I added some molecular work to try and understand whether there were any epigenetic signatures associated with that famine. And we generated some interesting data from that, but it was really eye-opening to me to visit Bangladesh in those days and see really what diabetes looks like in a South Asian population. And over the last few years, what I've been doing is building a very diverse diabetes research team at Queen Mary and working together particularly with colleagues Maniza Siddiqui, who I know many of you know, and Rohini Mathur, and really advancing our interest and our kind of excitement to really understand what diabetes looks like in diverse populations, particularly South Asians. And we're delighted to have a collaboration forming with Dr.Anjana and Dr.Mohan, who visited us only last year. So I'm going to talk a lot about the Genes and Health study, and this is a study that I co-lead with a colleague, Professor David Van Heel, and we've actually been going for 10 years now. Genes and Health is a study of British South Asians, really focused on improving the health of South Asian people in the UK. And why do we exist? Well, South Asian people in the UK experience significant health inequalities. And this is just data from our entire East London population who is registered with a family doctor. And what you can see just in that population of about 1 million people is that if you look at the prevalence of type 2 diabetes and the age at which that prevalence lies, you can see that people in their 30s and 40s, if you're white European, you've got a very low risk of having type 2 diabetes, but tenfold higher if you're British Bangladeshi. And 1 in 10 of our British Bangladeshi population in East London are living with diabetes. And by the time you get to your 70s, if you're South Asian, then there's a near inevitability that you or a close family member are going to have type 2 diabetes. So that's one of the underpinning reasons that Genes and Health exists. The second is that there is a huge lack of diversity in genetic research, and you probably can't see the very, very thin slice of green in this plot, which really just looks at the numbers of people recruited to genetic studies around the world. That green slice is South Asian people in those studies. It's appallingly low. And this is hugely important given that our genetics are very much determined by where our ancestors come from. And it really limits the potential for discovery, but also the transferability and generalizability of genetic studies to today. And South Asians form less than 1% of the total participants in the World Genetic Studies, despite being almost a quarter of the world's population. So Genes and Health is really a study that is built on understanding this huge lack of diversity and the health need of our South Asian population in the UK. And we're a large community-based research study that just aims to improve health across all disease areas through genetic research. We focus specifically on British Bangladeshi and British Pakistani people with a very clear justification. Those are the predominant South Asian populations in East London, but there's already actually increasing numbers of British Indians, but also Indians in India, enrolled in genetic studies, many of which are run locally. So we decided to focus on these two populations because there was particular need there. So we recruit individuals who are aged above 16, anybody and everybody that self-identifies as coming from one of those ethnic backgrounds. And our teams go out to community settings, GP surgeries, hospitals, mosques, anywhere that will have us. And to be recruited into Genes and Health takes a matter of about 10 minutes. We seek consent to link for our participants' health records held by the NHS, and I'll tell you more about that in a minute. We take a saliva sample for DNA, and then we also seek consent for us to be able to recontact our participants to invite them to what we call recall studies in the future. And those can be much more in-depth studies involving phenotyping, additional sample collection, and follow-up. We now work all over the UK, based out of sites in London, Bradford, Manchester, and West Midlands. And one of the reasons we've become a successful study is we're very engaged with the communities we represent, and our staff and the front face of the study is highly representative of those British Bangladeshi and British Pakistani people that we invite to join the study. And I made this slide a few days ago and was feeling very chuffed about our 66,982 volunteers, but I just got an email this morning saying we're at 70,000. So we're very proud of the fact that we've become a very large study, but not so proud that we can't reset our target constantly, and our current target is now 100,000. And we're a very large team now, both scientifically and operationally, probably not as large as MDRF, but this is just some of the key team members. And we have partnerships across academic institutions in the UK, the NHS, community organisations, and funding from a number of sources, including the Wellcome Trust, Medical Research Council, and NIHR. And over the last few years, we've also had a very, very positive life sciences consortium with 8 pharmaceutical partners who have funded a lot of our work and provided great scientific insights as well. So how does Genes and Health actually work? Well, we only spend 10 or 15 minutes with our participants when they sign up, but we have the most incredible resource of NHS health data that we securely link to, that provides a 10, 20, sometimes 30-year picture of their health. And that might be what we call binary traits, so does an individual have a disease and when did it start? But also quantitative traits, so blood test measures, blood pressures, weights, heights, all of the things that are measured in routine healthcare in the UK. And in total, we have over 3 million years 3 million life years of data on our volunteers, and that's purely because of this linkage to the NHS health data. We haven't had to collect any of that data. So, for example, for serum creatinine, we have nearly 1 million readings of creatinine for 44,000 of our volunteers, and these are just some trajectories of creatinine measures over the ages of certain individual volunteers, and you can see that really just by linking to this data, we can plot out trajectories and see who's developing renal failure, who's recovering from renal failure, etc. We then, from that saliva sample, have genetic data. We have both genotype chips on just over 50,000 of our volunteers using an Illumina chip, and that's been really made research ready by already being imputed and put in Regini-ready files. We have high-depth exome sequencing on 55,000 of our volunteers, which has gone through a lot of QC as well as annotation of rare variants, and we're beginning to build up a multi-omic data set, so far in just 1,700 of our volunteers, but we're hoping to take that to 10,000 over the next few years with multiple proteomic platforms, metabolomics and single-cell RNA-seq, and that's all from plasma samples. So with all of that data, what we've been able to do is become a global research resource, and we don't hold this data just for ourselves and our own research. We make this accessible to researchers around the world through a very simple access process, a little bit like applying to UK Biobank with a minimal fee, and we now have over 250 researchers working with our data, which is all held in a Google Cloud, highly secure, trusted research environment. And by setting up the study in this way, we've been able to really drive a lot of research, much of which is collaborative and led by external partners, and generate a very high impact collection of papers. And if you follow the QR link, you are welcome to photograph that, then that'll take you to our publications. So what I'm going to do now is talk through some of the diabetes research we've been doing in Genes and Health. And I should say this is very much team science, and I work really closely with Manisa and Rahini and all of our team members to drive a lot of kind of research to better understand how genetics can inform care, prevention, diagnosis of diabetes. So I'm going to first focus on some of the science we've been doing simply with just our genotype arrays. And for those of you that don't know much about genotyping, this is really now very cheap and accessible genetic technology. It costs less than $100 per sample, probably a lot less in India, where there's some great biotechs making these technologies. You only need to do a genotype once a year, once in a person's lifetime, because your genetics doesn't change. And the pipelines for generating data from genotype chips is very automated and rapid. So if you compare this to doing a blood test or a few blood tests, actually the cost is not hugely different, and the costs are coming down. So we've been working with our genotype data to ask a few questions. Firstly, around prediction of type 2 diabetes and gestational diabetes risk. Secondly, to help understand whether we can classify type 1 and type 2 diabetes better. Then to drill down on some South Asian specific type 2 diabetes etiology, and then to work with some new common genetic risk factors for type 2 diabetes that haven't been well described before. So I'll give you just a flavour of this. So first of all, we've been looking at how something called a polygenic risk score, which really just characterises the small amounts of genetic variation that we all hold across our genomes, not big mutations, just subtle small changes. And you can put all of that together into a polygenic risk score that then can be used to predict risk of any disease outcome you like, but we've worked on type 2 diabetes. So we built a polygenic risk score that was tuned specifically to the ancestral backgrounds of our South Asian population, and then we added it to a clinical risk tool that's commonly used by the NHS called Q diabetes. It's very similar to things like the Leicester score or the Cambridge score. And what we found is that by adding a genetic risk score to a clinical risk score, we can actually improve the prediction of who's going to get type 2 diabetes. It's not a huge improvement, but it improves particularly the prediction of risk in young people. And what we found is that 6% of young people where a clinical risk tool had determined that they were at low risk of developing type 2 diabetes, the addition of the genetic tool actually reclassified them as being at high risk. So there's potential utility of these kinds of tools if you ask the right question. And there's a lot of movement in the NHS to be bringing in these polygenic risk scores over the next few years. And just briefly, if you want to predict who's going to get gestational diabetes or who's going to move to type 2 diabetes after their gestational diabetes, you can use the same tools. The genetics of gestational diabetes are very, very similar to type 2 diabetes. So if you use a type 2 diabetes polygenic risk score, you can improve the prediction of both of these. So then we thought, well, hang on, what other questions might we be able to ask from these polygenic risk scores? And I'm sure many of you, and I certainly do in my practice, sometimes really struggle to tell the difference between type 1 and type 2 diabetes in younger, slim South Asian patients. So we thought, right, let's try and use these polygenic scores. There's one for type 1 diabetes, there's one for type 2 diabetes. We had to calibrate them to specific South Asian backgrounds. But by doing that with some very clever maths in between, we were able to estimate that 6% of our insulin-treated South Asian population in East London are classified as having type 2 diabetes, but in fact have type 1. This tool only works at a population scale at the moment, but what I'm really hoping is in the future, tools like this may really help us in clinical care to deal with these knotty clinical problems that we all face. So then what we did is we said, right, the polygenic score is just telling us whether it's somebody's at risk of having type 2 diabetes or not having type 2 diabetes, but there's all this literature around the heterogeneity of the condition that we all know. So it turns out you can partition a polygenic score into the underlying etiological pathways. And that's what we did with some reference studies to use. And what we were able to find then is that the genetic risk in our South Asian population in Genes and Health, the strongest genetic risk is arising from genetic risk to insulin deficiency and unfavorable fat distribution. Now, we see those things clinically, don't we? We've heard all sorts of people referencing these kind of descriptions of South Asian patients with diabetes. But what we've shown really is that there is a genetic risk for these. It's not all about what you're eating or what you're exercising or not exercising. There is a genetic risk to these specific etiologies and physical characteristics of type 2 diabetes that we all see. And if you put the people who are in the highest polygenic risk scores for those specific features, what we see in our Genes and Health population is that those individuals are diagnosed with type 2 diabetes 8 years earlier, they're slimmer, and they progress faster to retinopathy. So again, some genetic tools that are really giving us some helpful insights. And we also get a flavour of who's going to respond to what medications according to those genetic etiologies from the partition score. I won't go into great detail on this in the interest of time. The final piece of work just simply from our genotype data that we've done, and this is really important, I think, is to identify a common genetic risk factor for type 2 diabetes that's not previously well characterized. A lot of our British Bangladeshi and British Pakistani populations in East London still practice consanguinity within families, and in fact this is actually becoming more common in British Pakistanis. And what we were able to show by using a genetic tool and a bioinformatic tool that looks at what we call autozygosity, that's just the genetic readout of consanguinity across someone's genome, is that autozygosity is associated with an increased risk of various diseases, but one of which is type 2 diabetes. And at population level, we estimate that autozygosity accounts for between 5 and 18% of all of the type 2 diabetes cases we see in East London in British Pakistanis. A really big and important and very sensitive finding that we're now working through with our Community Advisory Board. And we also are trying to understand what it is from the autozygosity that's driving this additional risk, whether it's common genetic variation or rare genetic variation. I'm going to now just give a short flavour then of the work that we're doing using our exome sequencing data. And this, with an exome sequence, what you can do is you can delve much more deeply into an individual's genome, and in particular, you can characterize genetic variation that is rare or that is unknown. And that is really important in populations that are understudied in genetic studies, because you don't want to assume that the only discoveries that have been made in white European people are the only discoveries to make. Exome sequencing is a little bit more expensive. But it's increasingly automated, and actually the prices are coming down, and in 2 or 3 years they'll probably be around the same as genotyping costs. It is computationally a little bit more difficult though. So we've been doing a lot of work in Genes and Health on rare variants, and in particular rare variants where there is a loss of function in the gene. And because of the consanguinity in our volunteers, we see more commonly than most other studies homozygote individuals carrying loss of function gene variants, which are essentially we describe them as a gene knockout. It's where a gene is completely obliterated in terms of its function. And that really unlocks a whole load of new biological understanding, but it also tells us about drug mechanism. We're beginning to identify some novel type 2 diabetes etiology from doing this, as well as trying to improve the diagnosis of type 2 diabetes. So this isn't a diabetes story, but this is the first rare variant study I did, and I was a bit skeptical. I'm much more of a population health person, and I thought, can we really make that many discoveries from a single rare variant? And this is a rare variant in the MC3R gene, which is related to MC4R, which we know is an obesity gene. And a lot of people were trying to characterize what this gene does, but really not making much headway from population genetic studies, functional studies. Now, in Genes and Health, we had a single individual who had one of these gene knockouts. He was homozygous for a loss of function variant in MC3R. And by studying him, this one individual in my clinic in East London, we were able to work out exactly what this gene did, and then complement it by all of the population genetics that was there and functional data. And this unraveled a story about pubertal development and early growth and development. And that was the point at which I thought, okay, hang on, this rare stuff really is going to open our eyes to things. I've put this slide in for Professor A.Nauck. We've got some really interesting individuals in our study carrying gene variants and loss of function variants in incretin pathway genes. And I've spent the last 2 or 3 years with my team recalling some of these individuals, and in particular individuals, a small number of them, carrying loss of function variants in the homozygous state in the GIP receptor. And I don't need to articulate again because it's been done by someone who knows far more than I do earlier, but we've got this slight conundrum with GIPR, which is, do we agonize, do we antagonize? And the data is kind of pointing different ways, and tirzepatide has gone one way and the new Amgen drug is going the other way. So what we demonstrate with our homozygous individuals, but also our heterozygous individuals, is that they are, compared to age and sex matched individuals in Genes and Health, at lower body mass index. But what we've also then done over the last few years, and I can't share an awful lot of this data at the moment because it's a little bit sensitive, but we've essentially brought about 8 individuals in and some matched controls for mixed meal tests. And we've then been studying the impact of these GIPR knockouts on the response to a meal, and in particular GIPR incretin responses. And what we've discovered just very recently is we see a fasting and post-meal increase in a peptide hormone that is known to reduce appetite and food intake in experimental studies. And we're just taking that a lot further and hoping to have a publication on that later on in the year. And that's work in collaboration with Steve O'Reilly in Cambridge. Moving away from that into type 2 diabetes etiology, and what can we learn from rare variants there? And this is really work led by my wonderful colleague Maniza Siddiqui, and she's been the intellectual kind of powerhouse behind this. But we've essentially done something called an exome-wide association study, which looks in a very unbiased way across our exome sequences, comparing people with and without type 2 diabetes, to find novel rare variants associated with type 2 diabetes. And I'm going to tell you a little bit about one of our top hits, And you'll all recognize this gene name, it's HNF4 alpha, but it gets a bit more interesting. This is not MODY that we've discovered. Now, this specific variant that we've identified in HNF4 alpha is not well characterized before. In fact, it's never really come up in all the type 2 diabetes genomic studies. The reason being it's almost non-existent outside South Asia. It's somewhere between rare and common in South Asians. The allele frequency, I think, is about 8%. And if you look up in our kind of reference panels that predict the function of variants, this one comes up as a benign variant, so it's not known to cause monogenic diabetes. It gets more interesting. So then if we look at the carriers of this variant in Genes and Health, we have plenty of people who don't have the variant, but we have 886 people who are heterozygous and 13 homozygotes. Only one of those homozygotes has type 2 diabetes, and yet a quarter of our population in Genes and Health has type 2 diabetes. So we then ran this variant through what we call a PheWAS pipeline. That's really looking for the phenotypic, the clinical characteristics associated with single gene variants. And what we find is that this HNF4 alpha variant is actually protective over type 2 diabetes. So absolutely the opposite to the HNF4 alpha we see in monogenic diabetes. And that's correlated with a lower HbA1c. Interestingly, individuals carrying these variants have a higher BMI and waist circumference. But we have then also gone on to our proteomic data to say this is a gain of function variant, which is the opposite of the loss of function variants that are in monogenic diabetes. And we are absolutely delighted to be working with MDRF and many of you here in the room who have really kind of risen to the challenge of saying, well, Genes and Health is making some discoveries, but we need to work with other people to really shore up those discoveries and make sure that they are true, but also that they apply to wider South Asian populations, including Indians. And I won't go through everyone's names, many of you are in the room, but really a huge thank you for supporting this work and for our collaborations. And the first bit of replication we've done with MDRF with this HNF4 alpha variant, because there aren't many controls in MDRF, we haven't been able to look at the impact on type 2 diabetes, but certainly the signals that we're seeing in BMI, waist and weight are beautifully replicating our findings, which is fantastic, and we're doing more work on that at the moment. Okay. Then just the final story to convince you, if I haven't already, that genetics is really cool but also really important, is work that my team are now doing through a Wellcome Discovery Award, looking at the HbA1c blood test, which the NHS measures about 50 million times a year, and we now use this universally for both diagnosis and monitoring of diabetes. You all know that there are problems with HbA1c, particularly in people with anemia, thalassemia, sickle, G6PD deficiency. But there is an issue that we have because those conditions are rarer in our population, but also there are many people with clinically silent conditions, and that is impacting the accuracy of HbA1c. So we've been doing a lot of work on this. We've got a lot of work going on in G6PD deficiency with Inej Barroso in Exeter. We're starting some work on thalassemia. But in the process of all of our genetic studies in Genes and Health, we've identified another variant that's only common in South Asians called PISO1. And this is a red cell gene, but not very well characterized. And we discover that people carrying a single allele, a single variant in the PISO1 gene have an HbA1c that is 6 millimoles lower than people without the variant. You may say, okay, so what? Maybe that's associated with a lower glucose as well, but it's absolutely not. And this plot here, sorry, it's a bit small, just shows the discrepancy between HbA1c and glucose. And this is really an undetectable, clinically silent variant that is having a big impact on the accuracy of HbA1c. And then when we put this into some survival models, what you can see is people carrying the variants, you only need to be a heterozygote, and that's pretty common. It's about 4% of people in our population. Those individuals progress to retinopathy far more quickly than people without that variant. And that's because we are inaccurately diagnosing their type 2 diabetes late, but also offering them probably false reassurance that their HbA1c is normal when it's not. And this is work that my team are now doing a lot on PISA, thalassemia, and all the other ones, because this is a really, really important story for us all to tell, because we think it's leading to significant inequalities and poor outcomes. And we've got a new study just in setup at the moment called Black Health Legacy, which is going to be similar to Genes and Health, but in Black African and Black Caribbean people living in the UK. And this is really important because where those thalassemia variants are common in South Asians, in people of African and Caribbean ancestry, the G6PD variants are super common, and there's a really big story to tell there. So, in summary, I hope I've convinced you that genomics has the potential to inform many aspects of diabetes prevention and care, and that our advanced genetic and multi-omic technologies are generating a lot of new insights. It is vital that we look to South Asian populations for our research to have impact. And I hope, I'm preaching to the converted here, but I think this is really, really just emphasizing the need for initiatives like MDRF and all of the fantastic work you're doing and all the genomic data that you're collecting. This work is a long way from clinical translation, and particularly in resource-limited countries. But what I'm really hoping is that over the next few years we begin to see some trials, some health economic analyses, and some implementation research that really guides how we can use genomics cost-effectively and well. And just finally to say that there is no doubt that for big genetic studies and for any good research, partnerships and collaboration are critical. And I thank everybody at MDRF again for the invitation and also for the collaboration that we've had and we will continue to have going forward. Thank you very much.